Hello everyone, my name is Xin Lai, currently working at the University Hospital of Erlangen. I would first like to thank the committee for giving me this opportunity to present my research work, and the title of my work is Network and Systems-Based Engineering of Dendritic Cells with microRNAs for Cancer Immunotherapy. So first I would like to give a short introduction to dendritic cell and microRNA. The dendritic cell is a kind of antigen presenting cells. As you see in this plot, they can process and present antigens to immune cells like T cells, which enable them to kill the tumor cells. And microRNA is a non-coding RNA, and it can repress the gene expression at the post-transcription level. It is also known that they play a crucial role in regulating phenotypes of dendritic cells. So in our department, the experimental researchers have invented a kind of dendritic cells with superior immunogenic function, and such dendritic cells are being tested in clinical trials for treating melanoma patients. So we get samples from the donors, and from the blood, we can obtain monocyte-derived dendritic cells and then culture them with some proteins like cytokines to neutralize them. After getting the mutual dendritic cells, we treat them with Mena-A, which is the antigen for melanoma, and the RNA encoding the active IKK beta that triggers NF-kappa signaling pathway. Afterwards, we co-culture these CIA-KK dendritic cells with the T cells, and leads to the repetitive proliferation of the T cells over weeks and also a memory-like phenotypes of the T cells. This means that the T cells can efficiently recognize the melanoma cells and kill them. And from a systems perspective, we hypothesize that this superior dendritic cells phenotype is determined by complex gene regulatory networks. So as we assume that there is complex regulatory gene regulated networks regulating the immunogenetic function of dendritic cells, therefore in our study, we first want to reconstruct the gene regulatory networks underlying the immunogenic function of the dendritic cells and understand the role that microRNAs play in regulating the immunogenic function of the dendritic cells and identify microRNAs that can improve the immunogenic potency of the dendritic cells. So to do that, we apply a systems biology approach in our study, and the approach integrates computational methods and experiment data to investigate the microRNA function in dendritic cells, and I will explain these steps in details in the coming up slides. So first, we generate the transcript profiles of the dendritic cells using the RNA sequencing, and after processing the data, we identify dozens of different expressed microRNAs that can distinguish the two types of dendritic cells. Of note, we also have the expression profile of the protein coding gene, and this result suggests that the microRNAs play a crucial role in regulating immunogenetic function of the dendritic cells and requires further investigation. Then we perform Geneset enrichment analysis using the Reconstructing database. The database contains over 2,000 pathways in dozens of categories, which are highlighted by those labels as you see on this map. In total, we have identified 195 pathways, and most of them belong to the transduction and the immune system categories. Interestingly, 12 pathways are associated with NF-kappa activation and signaling. This fits very well to our expectation that, as we know, the IKK beta we put into the dendritic cells can activate the signaling pathways. Here I show you how we reconstruct the networks underlying the pathway categories that we have identified. To do so, we use the functional interactions between protein coding genes that are derived from the Reconstructing database. This includes activation, inhibition, complex formation, and co-expression. In addition, we have used other resources to derive the microRNA gene interactions, and these interactions are based on putative interactions with experimental validation. So we further filter those interactions with Pearson correlations to get reliable gene regulatory networks. As a result, we reconstruct the networks for each pathway categories, as you see here, with the size ranging from dozens of genes to almost 2,000 of genes. So after obtaining these networks, we want to combine them with the gene expression profiles to help us rank the genes. Here we make use of this idea called guillotine by association to identify crucial genes. It means the genes with high potential in their expression are important, and if their interacting genes are also largely perturbed, this makes the genes more important. So here is an illustration how we quantify the genes importance based on their expression perturbation and the network topology. And in these two cartoon networks, you have to see that the genes are in different colors, and the colors indicate the node weight of genes quantified by their expression statistics. And we transform the network into a map, and where the distance between the genes were characterized by their regulatory strengths. So which they are actually here the Pearson correlations. And for each gene, we calculate the score by starting from the minimum distance, which is 0, to the maximum distance, which is from the 0 to the furthest node in the network. And this results in a curve over a range of D, as you see in this plot. And then we use the errors under the curve to quantify the importance of the gene in the network. And the bigger the error is, and the more important a gene is. So if we compare gene J and gene K, the gene K is more closer to genes with high node weights. So it means it has a higher score, and which means it's more important. By applying this concept to the microRNAs, we say that microRNAs have a strong effect on many perturbed protein coding genes are more important in our networks. As we are particularly interested in how the microRNAs can regulate the immunogenic function of dendritic cells, we further draw the heat map to show the landscape of microRNA gene interactions in immune signaling pathways. In the upper part of this heat map, the gray grades show whether or not a gene belongs to an enriched pathway identified by us, and red framework highlight genes with annotated immune function. And in between, there's a bubble to show the log2 further change of the genes. In the lower part of the heat map, we show the microRNA gene interactions and their regulatory strengths quantified by Pearson correlation. And microRNAs are ranked using the network modeling method, with the most ones appearing on the top. Such a figure shows how microRNA gene interactions at the pathway level, and allowing experimentalists to select microRNA gene interactions of interest to do experiments. In addition, we have created a database to store the results that can be further used by other researchers. So finally, we want to link those microRNA gene interactions to immunogenic function of dendritic cells. We performed meta-analysis by searching experiment data that help us to comprehend the function of the microRNAs. Here, the diagram shows the relationship between microRNA gene interactions and the phenotypes of dendritic cells. The left column are microRNAs, the middle ones are protein coding genes, and the right one are the phenotypes of dendritic cells. In between, the lines are indicating the regulatory strengths between microRNAs and protein coding genes, and the lines are showing the association between protein coding genes and the DC function that are crucial for immune response. The arrows on the microRNAs indicate how we can modulate the microRNA expression to improve the immunogenic potency of the dendritic cells. Specifically, I want to highlight some top microRNAs identified by us using the network approach. For example, we have this microRNA 15A and 16 target IKK beta that can activate NF-kappa signaling pathway, regulating DC activation and maturation. And microRNA 144 regulates the MAPK signaling pathway by targeting MAP3K8, regulating the NF-kappa activation, which is also associated with T cell priming and activation. And finally, this microRNA 21 targets cytokines such as interleukin 12 and CCL20 that are important for DC-mediated chemotaxis. Finally, I want to summarize. So we have developed an approach that systematically identifies functional microRNA gene interactions in dendritic cells. We delineated the landscapes of microRNA gene interactions at the pathway level. In dendritic cells, we identified dozens of the promising microRNA candidates, such as microRNA 15A and 16, for improving the immunogenic potency of dendritic cells. So finally, we also create a database that allows for further experimental validation of the results. So in the end, I would like to thank my colleagues and collaborators who provide numerous support for this project, as well as the funding agency. And thank you very much for your attention. I would like to answer your questions.